Welcome everyone. Today we are going to see about DNA fingerprinting under Unit 5 of our DNA technology. Okay, coming under this topic, what we are going to discuss about is introduction and what is DNA fingerprinting and its working principle. Then what is satellite DNA? Then methodology of DNA fingerprinting and what are all its applications. Okay, let's see what is DNA fingerprinting. It is a process of obtaining fingerprint patterns of DNA on X-ray film photography. Simply, it means obtaining DNA images by using X-ray film photography as like thumb impression. Thumb impression uses ink or dye, but DNA fingerprinting uses several molecular techniques. So for example, isolation techniques. So there is isolation of DNA, then a PCR amplification methods, gel electrophoresis method, southern blotting method, then X-ray crystallography method. Hence, DNA fingerprinting is otherwise called molecular fingerprinting method or DNA profiling method. This technique is the first proposed by Alec Jeffreys. In animals, it is called a zoo blot. DNA fingerprinting done in animals, it is called a zoo blot. If DNA fingerprinting done in plants, it is called a garden blot. DNA print patterns is unique for an individual. How our fingerprints, thumb fingerprints is unique for individual. That does not match with anyone. As same like that, a DNA fingerprint also unique for an individual. It does not match with anyone unless very close relatives. Let's coming into the principle of working based on the presence of satellite DNA. It is otherwise called a junk DNA. Satellite DNA means non-coding regions of DNA which is present in between the coding sequences. For example, variable number of tandem repeats are present in further, that is for example CGCA, 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 which is repeatedly present in the non-coding region. Whereas in Sun, CGCA, CGCA, that is the same sequences but varies in length. It is called variable number of tandem repeats. DNA fingerprinting mainly focused on this variable number of tandem repeats present between the two individuals. Okay, then variable number of tandem repeats, simply it is called as a VNTR, which varies in length. Then what is mini satellited DNA means, which is a very small repeated sequences, mainly to have a only 9 to 40 base pair in size. So it is used as a marker key for DNA fingerprinting technique because DNA fingerprinting technique are fully focused on this type of sequences. So it is used as a marker key for this technique on these sequences. Okay, so it is represented as a marker key for doing the DNA fingerprinting technique. Next we see the methodology of DNA fingerprinting. Already I told you that DNA fingerprinting technique uses several molecular techniques. First we can isolate DNA, then we go for restriction digestion, then PCR amplification method, then next step is a gel electrophoresis method, then southern blotting technique, selection of DNA probe, hybridization technique, autoradiography, then analysis of DNA print patterns. So these are the several molecular techniques followed in DNA fingerprinting technique. Okay, let's see the methodology of DNA fingerprinting. In this technique, we can use several molecular techniques. First is the isolation of cell DNA. The DNA can be isolated from source organism, and DNA fingerprinting is majorly used in forensic science department. So that DNA can be isolated from suspected crime individuals. The sample may be blood or semen from rape victims, saliva, teeth, bone, tissues, and hair follicles, which is collected from crime place. Low amount of sample is enough for this technique. Then we go for restriction digestion. In restriction digestion, the sample DNA can be cut by using restriction enzymes into short DNA fragments. The short DNA fragments may be high molecular weight or low molecular weight DNA can be obtained. In that short DNA fragments, our mini satellites may also present. Then we go for PCR amplification method. In this method, our sample DNA is less in amount. So that we go for PCR amplification method. To make several copies of DNA using PCR machine, already we have less amount of samples. So we can multiply into many copies by using PCR machine. Simply amplification method, we can obtain million copies of DNA within few minutes. Next we do gel electrophoresis method. In this method, fragmented DNA sample is loaded on agarose gel electrophoresis. In this what will happen means DNA fragments get separated due to the molecular weight. Low molecular weight run faster and high molecular weight remains in the same place, so that DNA bands will appear. After the completion of gel electrophoresis, we go for southern blotting method. In this method, the electrophoresed gel was placed on blotting apparatus. Here what happened means transfer of a DNA bands to nylon membrane or nitrocellulose filter paper. And then nitrocellulose filter paper baked at a high temperature for about 55 degrees Celsius for few seconds. For the reason of DNA bands get fixed on the nylon membrane. Then selection of DNA probe. Alec Jeffries from USA constructed DNA probes for DNA fingerprinting method. Already you know that a probe means known sequences of DNA fragment already radio labeled with isotopes. But due to the high cost of DNA probes developed by Alec Jeffries, in India, Dr. Lalji Singh, who worked on a Center for Cellular and Molecular Biology department in Hyderabad, he developed DNA probes from banded grater, that is a poisonous snake. He developed a DNA probe for DNA fingerprinting method. Then hybridization method. In this method, a DNA probes, that is a known sequences of mini satellite DNA. This nitrocellulose filter paper placed in alkali solution to denature the duplex DNA. Then that denatured duplex DNA is placed in a hybridization solution containing DNA probes. Here, DNA probes get hybridized with appropriate mini satellites of source sample. Then wash unbound probes. Then we go for autoradiography. Here, filter paper placed in extra film for about 3 hours, so that the radioactivity of probe make dark spots on X-ray film. Then DNA fingerprint pattern shows. Then analysis of DNA print pattern. If all bands matched 100% with the suspected crime sample, that accused found to be victim. Let's see the sample picture. In this picture, our DNA sample collected from crime scene and also from some suspected individuals. In that, what we can see is the DNA sample obtained from crime place will be perfectly matched with suspected crime individual 2, so that the suspected crime individual 2 is identified as a victim. In this picture, we see the overall method of DNA fingerprinting. First of all, we can isolate the DNA from a source organism or from crime places. Then we extract the DNA by using several techniques, for example phenochloroform extraction method and so on. Then we go for restriction digestion. Here we use restriction enzymes which can cut the DNA fragments into short DNA fragments. Then we go for electrophoresis method. In this electrophoresis, the DNA bands can be obtained. Then we go for a southern blotting method. In that place, our DNA fragments can be transferred to nylon membrane or nitrocellulose filter paper. Then we go for hybridization technique where DNA probes get hybridized with our DNA fragments. At last, autoradiography. In that our DNA fragments get fluoresced on X-ray film photography due to radioactivity of probe, so that dark spots may be appear. Finally, we analyze the DNA fingerprint patterns. Then let's see the applications of DNA fingerprinting. First of all, it is majorly used in forensic science department to detect rapist, murderer and some unknown death cases. And it is also used to detect some immigrants, those people who are migrated from other countries. To detect them, it is used. Then it is also used to detect the disputed parentage. Then it is used to trace some murder cases and rapist, and is used in pedigree analysis and some genetic analysis for research purposes. In India, Center for DNA Fingerprint under Diagnostics, who conducted that sample forwarded only from court or police. I hope that you all understand this topic, and thank you for watching this video. Thank you so much.